brings me to the final speaker of this session. His name is Adam Majcher. I hope I said it correctly. He did his PhD at the University Hospital of Zurich. He has a background in lipid pathobiology, multi-omics in a translational fashion. I'm going to keep going. Biomarker discovery. And I think currently you're at the LUMC, but also working with Boeringer in Ingelheim, right? I butchered that. And as well, I think that's it. Yes. Good luck.
Thank you very much. It's really
oh, no, you're going there. Yeah.
Oh, this is complicated. So hello everyone. You might not see me. I might be too short for this country, but yeah, sorry for that. So hello, my name is Adam. I believe I'm the guy keeping you from the coffee break, but I promise I will try to make it count. So I work at the LUMC, but I joined quite recently, so most of the work I will show is actually from my time in Switzerland. So I'll try to tell you a little bit about type 2 diabetes neuropathy and multi-omics approaches. So, not this one. Oh, perfect. So, type 2 diabetes, very active field. I might have to change these numbers because of all the Ozempic and stuff. But what is important is that about 50% of diabetic patients develop peripheral neuropathy. What it is progressive loss of sensory neurons in your skin. And it's actually quite a bad complication that can lead to ulcers and even amputations. And the issue is we don't really have a good biomarkers nor direct therapies for this besides adjusting the risk factors. So we grouped up with Karolinska Institute in Sweden, and we got this nice cohort of almost 100 patients, age and sex matched with diabetes, and we wanted to perform multi-omics analysis. So this has been done many, many times, but what is special here that we did not only, oh, can I go back? Yes. So not only collected plasma, but we also collected skin biopsies. This is actually quite hard to get because these patients actually suffer from wound healing because they have diabetes. And why skin, you might ask, is because that's where neuropathy starts. So then, when we had this, we performed at the Unispital Zurich lipidomics, metabolomics analysis, at the ETH proteomics analysis together with Picotti lab. So now, when we looked at the lipids, we realized something very surprising, that we have increase in one specific class of sphingolipids. And these are atypical sphingolipids. All these red dots belong to the class of 1-deoxysphingolipids. These are actually quite known to mediate neuropathies. And what was even nicer, when we stratified the patients, we realized that patients that had already developed neuropathy and neuropathic syndrome, they had even increased deoxysphingolipids in their skin. So we got very interested in this class, but I believe we should step back a little bit, and I should introduce what sphingolipids actually are. So sphingolipids, my favorite class of lipids, but you might ask, why are they so special? Why we don't call them just lipids, but sphingolipids? So what's different with sphingolipids? It's that they are actually based on a long-chain base instead of just glycerol. So what is the long-chain base? Long-chain base is actually product of condensation of amino acid, usually L-serine, and activated fatty acids. And this first step is catalyzed by the enzyme called SPT, or serine palmitoyl transferase, that makes our long-chain base, and then we continue to the pathway. But under specific conditions, SPT can take another substrate, which is L-alanine. And now, I mean, you don't have to be expert chemist to see what's the difference between serine and alanine. It's this hydroxy group. So if you utilize L-alanine, you're missing this hydroxy group. Therefore, you're making 1-deoxysphingolipids. And we know that under certain conditions associated with neuropathies, such as rare neuropathies like hereditary sensory autonomic neuropathy type 1 or HSN1, SPT loses specificity towards L-serine, uses alanine, increases deoxysphingolipids, and as you can see here in DRGs, dorsal root ganglia, the sensory neurons are really, really not happy about deoxysphingolipids. But now a very important scientific question, why? why do we have increased 1-deoxysphingolipids in diabetes? We don't have any mutations or associated with sphingolipid pathway and type 2 diabetes. We have it in MACTEL or HSN1 or other neuropathies, but the reason for increased diabetes 1-deoxysphingolipids is not very understood. So we went further and we wanted to investigate, and I really like stable-isotope tracing. We already heard a little bit about it. So what we can do, we can just supply these labeled amino acids, measure lipidomics, and check how much we're producing. So I had this idea during my PhD, what if I just don't do much and just put a lot of alanine on cells? You can see that, yeah, if you increase alanine, you just start forming a lot of deoxysphingolipids. But what was interesting is that even canonical sphingolipids, so serine products, went down. So I had this idea, what if it's not the total volume? What if it is actually a ratio between two amino acids, so alanine to serine ratio? And now how can you prove ratio in biochemistry? So ratio can be either alanine going up or serine going down. And with stable-isotope tracing, you can design this experiment where you're increasing alanine or decreasing serine. And then when we measured the fluxes of sphingolipids, we saw that, oh, I might have discovered something interesting. This is very, very, very linear and reproducible. So it doesn't really matter if I'm increasing alanine or decreasing serine. As long as I'm increasing alanine to serine ratio, I'm increasing deoxysphingolipids. So then we went back to our cohort, measured amino acids, and we see that, okay, is it alanine going up or serine going down? It's actually both. So we have increased alanine, decreased serine, and increased alanine to serine ratio. And is this somehow associated with deoxysphingolipids? So we took another cohort of patients, almost 700 people, measured amino acids and deoxysphingolipids, and we found some association. But this doesn't really answer the question, right? Why is alanine to serine ratio increased in diabetes? So how can we study these things? So I really like stable-isotope tracing in metabolic research. So for example, what you can do, you can exchange the substrate of glucose for heavy glucose, so all carbons are C13. And then you see that glucose is actually precursor of both alanine and serine, therefore 1-deoxysphingolipids and canonical sphingolipids. And if you do this and resolve it in a time-dependent matter or different concentrations, or you measure untargeted or targeted metabolomics, lipidomics, you can really nicely start investigating what's going on and why do we have increased alanine to serine ratio. For example, first catch, how can be alanine increase? So we have this very famous enzyme. I believe in Netherlands, if you have too much Heineken, your GP might look at your ALT. So this stands for alanine aminotransferase, very famous liver enzyme. And this is an enzyme responsible for transamination and creation of alanine. So, okay, can this be our target? So we just knock down the ALT, and we see that indeed we are influencing the deoxysphingolipid production. So now, is this responsible? Is this a new target how to prevent formation of deoxysphingolipids? So when we took any of the rodent models, so these are rat livers with streptozotocin or high-fat diet mice or DB mice, and we took their livers and measured their metabolic activity of this ALT, we see that it's very, very, very increased, which makes it actually really interesting target. And we also know that if you knock it down, you protect animals. So I will not bore you more with very in-depth biochemistry, but basically you can go way further with this. So you can ask question, okay, if deoxys are coming from alanine, and alanine is coming through ALT from glutamate, where is glutamate coming from? From glutamine. Where is glutamine coming from? We actually managed to track it down to even protein degradation, especially associated with diabetic sarcopenia, and this is directly associated with insulin receptor. And proteasome to deoxysphingolipids, that's kind of far-fetched, but it works even in animals. And we're also glad to see our proteomics results where the most increased pathway was actually proteasomal complex. But now, I want to really switch gears because, yeah, we did a lot for the setting side, but I think we should start thinking in multi-omics a little bit like this. Can we actually use this data for new therapies? Can we make use and actually deliver for our patients? So you might argue, okay, if you're claiming that alanine to serine ratio is somehow important in diabetic neuropathy, there is an easy way how to decrease it, right? So you can just increase serine. Serine is amino acid, but you can just supplement it. So this is what we did. If you take animals again, you induce diabetes, you have increased alanine to serine ratio, therefore you increase deoxysphingolipids, and then if you feed them L-serine, you see alanine to serine ratio drops, deoxysphingolipids drops, and these animals are protected from neuropathy. So this is quite nice, and it's not just us claiming that serine is good. There were another two groups publishing the same results and same conclusions in actually quite nice journals. So this is quite reproducible. What about clinical trials? So far we have 2 clinical trials, unfortunately not on diabetic patients, but on these rare neuropathy patients, HSN1 patients, and we have some data suggesting this might actually work. But can we go further? So L-serine has its problems, mainly dosages. We can discuss it later. But now there is another technique quite popular, and that's CRISPR screen. So how does it work? You have your CRISPR expressing cells, you transfect it with the library and apply your toxin. This would be deoxysphingolipids, then you sequence. So when we did that, we actually found 5 genes that are responsible for this toxicity, and they all do the same thing. So if you look at this, this is very long-chain fatty acid synthesis, especially ELOF1, the elongase that synthesizes 2 main fatty acids, and that's fatty acid 24-0 and 24-1. This makes even more sense in the context of sphingolipids, because if you look where ELOF1 lies with regards to deoxysphingolipids, it's actually next to ceramide synthase 2, and this is the enzyme that takes our alanine product and attaches another fatty acids, therefore creating 1-deoxydihydroceramide. And this was very nice when I saw it, because I'm looking for a biomarker for neuropathy, and fatty acid 24-1 has a trivial name of nervonic acid, so that would be very nice parallel. So we proven it, and that was nice, but then I found these 2 papers from Vertex Pharmaceuticals that they developed ELOF inhibitors for XALD, and I was like, oh, this is nice, because they actually proved it in primates already. So we took these inhibitors, can these actually work and protect from the deoxysphingolipid-induced toxicity? So here you see very long-chain deoxydihydroceramides. If you treat isotopically labeled deoxysphinganine and then your inhibitor, ELOF1 inhibitor, or compound 22, you see that 24-1 nervonoic acid or lignoceric acid are going down, and then we see that cells are completely protected from the toxicity. So we can put deoxysphingolipids on top, but they just don't do their toxicity. And even nicer when you look at the sensory neurons, here these are dorsal DRG-derived sensory neurons. We see that they tolerate very nicely ELOF inhibition. When you put even 5 nanomolar deoxysphingolipids, you basically wipe out all the sensory neurons. And then if you treat with ELOF inhibitor, the toxicity is gone. So we can discuss where we put it there, but this was a very, very nice story that we managed to get out a few months ago. And now, for the last part, what we did not discuss, at least I didn't hear today, was GWAS and lipidomics association. So what it is, so we have genome-wide association studies. How does it work? You take a couple of patients, take their blood, you sequence, you run your panel, and then you try to associate these 2. And when we did that, we actually see that we found 2 hits, 2 enzymes. One is fatty acid desaturase 3, and then CYP4F2. And then now it's, I'm almost finished because it's very nice and simple in the end. We figured out that my colleague in Zurich, Gergely Karshai, characterized that basically FATS3 introduces this double bond on a kind of a strange position of 14-15, so all to my lipidomic folks check your double bond position, it's very important. And you see that when we introduce this double bond on the 14-15 position, deoxysphingolipids are not toxic anymore. So we can talk about first detoxification mechanism that was unknown, and it can be used further. And then what about the CYP4F2? CYP4F2 is known as omega hydroxylase of fatty acids. So the obvious question was, okay, can we omega, so at the back, hydroxylate deoxysphingolipids? And you see if you overexpress CYP4F2 and only CYP4F2, you start forming this omega hydroxy 1-deoxysphinganine. It's a quite complicated name, but it's aesthetically quite pretty. And what was super nice is when we synthesized this product, we see that omega hydroxy deoxysphinganine or deoxysphingolipids are not toxic anymore. So we have another very nice mechanism of detoxification coming from jivas. And then just to tell you in the end, if you synthesize, don't be afraid to look at your untargeted data, because we managed to supply this omega doxa, and we found at least 6 new classes of lipids and new metabolism, but I don't want to bore you. So now, did we manage to at least push a little bit the field that I leave up to you? But at least we know now that we have a couple of strategies how to tackle deoxysphingolipids and associated neuropathies, retinopathies, and wound healing issues, and that's decrease in alanine to serine ratio, decrease in the activity of ELOF1 and SIRS2 with inhibitor that is actually being developed, and then we can increase the detoxification. So just at the end, we are in the era of multi-omics. I mean, this festival is actually proof of that. So please think about your projects in a multi-omics fashion and be very careful with the data. But I believe that we are in an era where if you combine all the omics, you can create something we used to call digital fingerprint, and you can find very, very nice things. And then what am I actually doing in Netherlands? So I joined Martin Guerra's group, where I actually developed a platform for primary metabolism screening, also with stable-isotope tracing opportunities, data analysis, and flux. So if you have interesting projects that you would want to use for the metabolic studies, or you would want some data from us, please contact me or directly Martin. And if you like deoxysphingolipids or sphingolipids in general, you can contact Torsten or me, and I will redirect you. So yeah, thanks a lot.